Bacteria multiply by a simple form of cell division called binary fission. Using this process, bacteria colonies are able to grow. Binary fission is different to cell division in eukaryotes. This is because prokaryotes carry out binary fission, and these lack membrane-enclosed organelles like the nucleus. What conditions do bacteria need to reproduce? The purpose of cell division in bacteria is reproduction, which increases the population size. To reproduce, bacteria need nutrients. These nutrients are quite familiar to us. They are water, sugars, and salts. So if they have all of these things, how quickly can bacteria reproduce? If bacteria have enough nutrients and a suitable temperature, they can divide as often as once every 20 minutes. So here's our initial bacterium at 0 minutes. We've got just one. Now after 20 minutes, this one bacterium has divided into 2 bacteria. After 40 minutes, these 2 bacteria have divided again into 4 bacteria. And after another 20 minutes, these 4 bacteria have divided to produce 8 bacteria. So in the space of just 60 minutes, our bacterium has divided to produce 8 individual bacteria. So bacteria can go on dividing, but population growth will slow down or even stop if they run out of nutrients. How can we culture microorganisms like bacteria in a lab? Bacteria can be grown in a nutrient broth solution or as colonies on an agar gel plate. So on the left here, you can see the nutrient broth solution. This is a liquid that contains all the nutrients that bacteria need to grow successfully, such as carbohydrates for energy. And on the right is the agar plate. This is a solid gel with nutrients that also help with bacterial growth. It's important to ensure that the cultures grown on each of these media are not contaminated by other types of bacteria. We can tell that bacteria are growing because the nutrient broth solution will turn cloudy and the agar plate will start to develop individual colonies, showing the bacterial growth. Aseptic techniques are procedures used to prevent the contamination of pure cultures of microorganisms. Remember that microorganisms are small organisms like bacteria, and a pure culture is a collection of bacteria containing only one type of microorganism. In this diagram, you can see a petri dish. Growing inside this petri dish is a contaminated culture. This is a culture where different types of microorganisms have entered from the air and started growing. A contaminated culture like this one would confuse experiments. We might use pure cultures to investigate the action of disinfectants and antibiotics. It's really important that the culture is pure. For example, if we're testing how well an antibiotic works against a specific bacteria, then contamination would lead to inaccurate results. So how do we prepare an uncontaminated culture using aseptic techniques? First, petri dishes and culture media must be sterilised before use. In this diagram, you can see an autoclave. Equipment can be placed inside the autoclave, where it's then heated and put under pressure to destroy any bacteria. Pre-sterilised plastic petri dishes could also be bought. For the exam, you are expected to be able to explain why petri dishes and culture media must be sterilised before use. What other methods can we use to sterilise equipment? The inoculating loop used to transfer microorganisms to the media must be sterilised by passing it through a flame. In this diagram, you can see the inoculating loop being held next to a Bunsen burner. The flame kills any bacteria on the loop when it passes through. The loop is then allowed to cool down completely. This vial contains our sample of bacteria. When we open this to dip the inoculating loop in, we only remove the lid slightly. This is to prevent any bacteria or other microorganisms in the air from getting in and contaminating the sample. To further prevent contamination, we also flame the bottleneck. Now that we've done this, and once the inoculating loop has cooled down, we're going to dip the sterile loop into the sample. For the exam, you are expected to be able to explain why the inoculating loop must be sterilised before use. How do we now transfer the sample of bacteria to the growth media? The sterile inoculating loop is used to transfer the bacteria to the media by streaking. The media in this case will be agar jelly inside a Petri dish. To transfer the bacterial sample from the inoculating loop onto the agar jelly, we're going to streak it like this. You have to take care to make sure that you don't dig out or gouge the agar jelly while doing this. What's the next step? The inoculating loop is passed through the flame again before being used to spread the bacteria out further. So we pass the loop through the flame again, allowing it to cool down before being used for further streaks. The flaming kills the bacteria on the loop, and spreading it further dilutes the bacteria across the Petri dish. We pass the loop through the flame each time to kill any residual bacteria. Once we've done this final streak across the dish, we're going to pass the loop through the flame one last time. By preparing a Petri dish with a bacterial sample in this way, we're ensuring a dilution of bacteria across the plate. So there's a higher concentration of bacteria in this area where we did the first streak, and a lower concentration around here where we did the final streak. How do we store our Petri dish safely? The lid of the Petri dish should be secured with adhesive tape. You can see in this diagram that the tape has been placed in 4 places around the edge of the Petri dish. We place the tape in this way to prevent the dish from opening and also getting contaminated with other bacteria. It's also important that the tape isn't all the way around the edge, as this may prevent the growth or even promote the culturing of dangerous anaerobic bacteria. It's also a good idea to label the Petri dish with the culture that you're growing. The Petri dish should be stored upside down. So in this example, this top image is incorrect. We want the Petri dish to look like this. We store the Petri dish in this way to prevent any contamination that may form on the lid from dripping down onto the bacteria growing below. In the exam, you will be expected to be able to explain why Petri dish lids should be secured with sticky tape and stored upside down. So what do we do with our inoculated Petri dishes now? Cultures should typically be incubated at 25 degrees Celsius in school laboratories. This diagram shows an incubator. So we're going to incubate the culture at 25 degrees C inside an incubator like this one. We want the temperature to be below human body temperature to reduce the risk of culturing harmful microbes, but hospital labs may incubate at higher temperatures to quickly identify pathogens. You are expected to be able to explain why cultures are incubated at 25 degrees Celsius in school labs for your exam. What will our Petri dishes look like after incubation? As they are incubated, bacterial colonies will grow. The growth patterns that we'll see should look roughly like this. You can see how the bacterial growth has followed the pattern of our streaking. Thanks to the dilution that our streaking method achieved, we can see isolated colonies.
Thanks for watching. If you want to take your GCSE revision to the next level, head over to launchpadlearning.com and check out our smart learning platform that's been designed to get you top results in your exams. We cover your whole specification and make revision fun with interactive quizzes, easy to follow videos and more. You'll be kept motivated by your own AI tutor who's here to support you every step of the way. To check it out for yourself, click here. Or click here to keep watching a selection of the videos from our full GCSE biology course. See you there!